Hello everyone, welcome back to another session of RNA biology, and we were here in the previous class that we were trying to understand the importance of RNA decay, because RNA decay is as important as the RNA production itself and it has to be monitored stringently. We also should understand one more thing: if an important RNA is being formed in the cell and it is marked for degradation, then the system suffers, the cell suffers. So that is another thing, that RNA degradation is important, but premature degradation or unwanted degradation also should be prevented. But these things keep happening in a cell. So the adequate monitoring system, adequate surveillance system is important for maintaining the healthy quantity and quality of the RNA population inside a cell. Let us see RNA decay factor. This is a complex slide. Don't worry about it, because our purpose is not to go into detail. Of course, you can see these factors in detail, but the important thing is to sensitize you that the RNA decay factors, the mRNA decay factors, are not just one or not two or not three or not four. There are so many are there. They are in hundreds, and a lot of them are listed here. And understand, many more are not discovered yet, because we understand some mRNA population and their degradation, but we don't even know how many mRNA population we have in a human body. That itself is an enigma. We don't know how many genes constitute a human being. We don't even know an arbitrary number. We have only a guessed number, that we say around 1 lakh genes mRNAs are needed. That is anyone's guess. It's a ballpark figure. So it will take lot of years of research to understand the entire mRNA decay machinery. But whatever we have listed in this table, we will be trying to address the important members of this list, and we also try to understand what is the mechanism. And there are different categorization also exist for the mRNA decay pathway. So one category is deadenylation dependent mRNA decay, and name itself says that poly A tail is there, which is made of adenine, and adenine tail, if it is removed or degraded or lysed, then we call it a deadenylation. So the removal of poly A tail from the mRNA is called deadenylation. So deadenylation can trigger an RNA decay. So that is deadenylation dependent. And another example is deadenylation independent. That means the RNA can still have the poly A tail intact, but still it can be marked for degradation. So we call it as deadenylation independent. Then comes the third category, endoribonuclease mediated mRNA decay. Like we saw, nucleases can act right in the middle of the RNA, and it can cause creation of two ends, 5' end and 3' end, which is not protected. Usually the 5' end is supposed to be protected by 7-methyl cap, but you created by cutting right in the middle of the RNA. So that can have vulnerability to exonucleases. And also 3' end, new 3' end is created because of the endonucleolytic cleavage, and that is not polyadenylated. So that is also vulnerable. So deadenylation dependent mRNA decay, bulk of mRNAs undergo decay by this pathway. A majority, bulk means a good number of the mRNA population undergoes degradation by this pathway. The 5' 3' exonuclease pathway kicks in on top of that, and the exosome mediated 3' 5' exonuclease pathway also kicks in. So once deadenylation has happened, it is vulnerable to both kinds of exonuclease activity, 5' to 3' exonuclease and 3' to 5' exonuclease. Of course, you may wonder, oh, cap is there, what happened to cap? Of course, the cap will be removed by decapping enzymes. So if it is just like, if you see National Geographic channel, if you are watching, you will see in a herd, normally you see if there are a herd of buffalo, which is 100 or 200 buffaloes are there, if your lions are attacking them, they will be just charging onto them. They will just go after them. In this process, they will create a panic. And you may wonder why they have to do this futile exercise. They are searching for the weak. When they chase a herd, the weak one will linger, weak one will struggle. And they will fall back, or they will start showing signs of weakness. Same logic applies. If an mRNA's cap is removed, then it is a free for all. Other nucleus will jump in, remove the other parts, like it can, if the cap is removed or if the poly A tail is removed, then immediately the decapping enzymes will come, just like how lions will go after a weak animal. Same logic applies when it comes to enzymatic degradation of the RNA. And the deadenylation independent means poly A tail is intact, but still some specific decapping enzymes come into picture, and it can cause the degradation of the RNA population. And even if both these ends are intact, endonuclease can come right in the middle and cause the degradation. So let us see deadenylation dependent mRNA decay. How does it work? Shortening of poly A tail by deadenylation enzymes. Like I told you, many enzymes are capable of causing removal of the poly A tail, and we call it as deadenylation. And if the poly A tail has fallen below certain threshold, the PABP is no more able to bind. PABP need a certain stretch of poly A tail in order to bind. If the deadenylation happened, then PABP will not find the target attractive and it becomes a naked RNA, and this can cause deadenylation enzyme acting on it, and it will reduce. It will not cause the degradation, it will simply get rid of the poly A tail. Just like poly A polymerase was adding the A tail, the deadenylating enzymes will remove the poly A tail sequentially, and this step is reversible. Why? You can have cytoplasmic poly A polymerase. Just like you fall sick doesn't mean that it is a death sentence for you. You are sick now, you are not well, you will go to hospital, you will get a saline drip or maybe medicines, and you will back up and running. So same way, sometimes deadenylation happen, and if poly A polymerase recognizes in time and it realizes this is an important RNA, and then the poly A polymerase will keep adding the poly A tail onto the reduced tail bearing mRNA, and it will now become longer and it will back to normalcy, just like how you go to hospital and get recovered from your ailment. Followed by either removal of 5' cap by the decapping machinery and 5' 3' decay or 3' 5' decay. This reversible process did not happen. Say deadenylation happened, repair did not happen. Just like you are sick, you did not take medicine, did not go to hospital, situation can worsen and it can be life threatening. So we have to be careful enough to go to hospital in time. Same way, if an RNA is problematic, then you have to take care of the system effectively. That is why people who get some fever, some ailment like typhoid or maybe chicken pox, etc., after that they realize, after that they are losing lots of their hair, their skin has wrinkles, they are not well, they are weak, etc. Why? Because the body is using a lot of its resources in its immunity or fighting the virus. So the normal procedure like growing hair more, which is, I'm not saying hair is an unimportant part, but hair is a outcome of the biology of your cell, your epithelial cell in certain part of your body. Of course, it has got its own role like protecting from cold, your head has to be kept warm, especially in cold conditions. So hair does this insulation function. Many things we can discuss on and on about it. But when you are sick, body is not able to handle those so-called unimportant RNA population, then there is no repair happens. There is no those RNA responsible for hair growth may not get this helping hand of extra poly A polymerase. There is hardly any poly A polymerase available because they are running short. So they will not get extended and some proteins will go missing, and that will result in hair loss. This happens, all of you would have seen. Even pregnant mothers after their delivery, once their body is demanding a lot in the milk production, etc. So a mother's body is literally spending lot of energy in the uplifting or upbringing of the newborn baby. So her body is suffering from nutrition. So they also will face hair loss. I gave an example like this because this doesn't mean that your brain function can be compromised, or your digestion, or some of your heart beating, etc. can be compromised. So they are vital functions, they will be retained. So called unimportant functions will be like maintaining a strong, say you had a strong muscle and you have to daily do exercise and eat good protein rich food, etc. to maintain it. But if your body is little bit suffering, or body is wanting urgently energy for some other source, then your muscles will start becoming loosened muscles. You will have loose skin, saggy skin, etc. because body is not spending enough energy to retain the production of certain proteins required in those tissues by facilitating the rejuvenation of some defective RNA or a prematurely dying RNA. They are not repaired effectively because body don't have energy, it is being utilized for something else. So we have to understand deadenylation dependent mRNA decay. Although it is reversible, it need not be reversible. If it is not reversible, the polyadenylation or poly-A tail is not reversed, then it will be marked by removal of the 5' cap, and this itself can trigger the 5' 3' exonuclease, and the absence of poly-A tail will trigger a 3' to 5' exonuclease activity. So deadenylation dependent mRNA decay, we can see something like this. You have a mRNA, proper ORF, 5' untranslated region and 7-methyl guanosine cap, and you have a 3' UTR and the poly-A tail. This, since it is a cartoon, don't think that poly-A tail is only 4 A's. For convenience, 4 A's are shown here. If the deadenylation happened, by whom? By an enzyme called CCR4 NOT, or also called as PARN. So these are enzyme PARN, poly-A specific ribonuclease, that is the short form, and CCR4 NOT is the name of another enzyme that is capable of taking down the poly-A tail or reducing the length. So if this happened, then it can follow two track. One is 5' 3' exonuclease activity, that is starting from the 5' end, or 3' 5' exonuclease activity. How does it happen? Once this poly-A tail is shortened, then this can be bound by LSM 1 to 7. It is a ring protein. LSM 1, LSM 2, LSM 3, like that. LSM 1 to 7 proteins form a ring around the UTR, untranslated region, and this can trigger the decapping. Once this LSM 1 to 7 protein, just like I told you, a herd of buffalo, one buffalo is limbing. That is the dinner bell for the lion which is chasing it. So it can easily catch, oh, if I perceive little bit, this buffalo is my food. So this LSM 1 to 7 is just like a signature for the decapping process. This will trigger the decapping, and decapping is done by enzyme DCP2 and DCP1. DCP1 and DCP2 are two enzymes complex that will cause the 7-methyl guanosine to be lost. Now this will invite a 5' 3' exonuclease such as XRN1. These names of these enzymes are very important. If you do not understand the names of these enzymes, you will not understand the decay of the RNA. So XRN1 is a 5' 3' exonuclease, whereas exosome, we saw in one of the earlier class also about exosome. But say for example, LSM 1 to 7 did not bind for whatsoever reason, depending upon the UTR structure, secondary structure. Let us not go into the details what made LSM 1 to bind onto this deadenylated untranslated region of an mRNA. That is a different topic altogether. But understand, not every species of RNA which is devoid of poly-A tail will attract LSM 1 to 7. Some population attract LSM 1 to 7, which will cause the decapping eventually. But whereas if LSM 1 to 7 did not bind, does that mean the RNA will be safe? The answer is no, because the polyadenylation is important. So what will happen if the poly-A tail is reduced and cap is intact? but this in turn will attract the exosome. Exosomes are a group of 3' to 5' exonucleases, and that can eventually cause a scavenger decapping also. That means enzymes such as DCPS. DCPS is S stands for scavenger. Here we saw DCP1 and DCP2. So exosome itself is capable of doing the 3' to 5' exonuclease, but this will be further aggravated, just like putting salt in the wound. So it will further exacerbate the situation by introduction of DCPS, which is a scavenging enzyme, scavenger decapping, that will start the 5'. Then it is okay for XRN1 also, because once the decapping has happened or the 7-methyl guanosine is removed, then XRN1 can start acting on it. So whichever situation, whether it is via DCP1-2 complex or via DCPS, the decapping, if it has happened, it is an attractive substrate for XRN1 exonuclease 5', 3' exonuclease activity. Whereas if exosome directly coming into picture, it will degrade by itself. It do not need the help of any other protein, because exosome itself is a exonuclease. It will cause the 3' to 5' exonuclease activity. Let us see how these processes are explained in word. So this we are talking deadenylation dependent mRNA decay. So shortening of poly-A tail by deadenylation enzymes such as CCR4 or PARN. So poly-A specific ribonuclease, that is PARN. And removal of 5' cap by the decapping machinery, DCP1-2 complex. And then LSM 1-7 complex induces decapping by associating itself with the 3' deadenylating end. So this is another requirement for the LSM 1-7 mediated decapping. Although they do not cause the decapping, but their presence will attract the DCP1 and 2 to the 5' end of those RNA. After decapping, the 5' end no longer protected, and 5', 3' exonuclease, exoribonuclease XRN1, and that degrades the mRNA completely from 5' end of the mRNA. Now let us see deadenylation independent mRNA decay. In this, how does it happen? So it requires direct removal of 7-methyl guanosine cap from the 5' end of the mRNA. Means it will not bother whether 3' end is intact, 3' end is useful, functional, nothing. It will not bother. Here also the decapping enzyme is DCP1 and DCP2, same as where you saw LSM 1-7 was binding onto a deadenylated poly-A tail. However, here the DCP1 and 2 is not looking for LSM 1-7 because poly-A tail is intact. But here what happens, although an RNA that has got poly-A tail intact, the binding of two proteins, that is RPS28B and EDC3, these complex, or this is a heterodimer, if they are bound between the coding region and the poly-A tail or polyadenylated region, this we call it as 3' untranslated region. If EDC3 and RPS28B heterodimer binds onto this UTR, that can attract, just like how LSM 1-7 attracted DCP1, DCP2 complex. Similarly, this can attract this molecule, and this in turn will remove the 7-methyl guanosine cap, which makes the 5' end vulnerable to XRN1 exonuclease. So this is basically called the EDC3 protein's name is enhancer of decapping. That means EDC3, when it is found, it will attract the DCP1, DCP2 complex, and the moment DCP1, DCP2 complex that has come to the 5' end of an RNA, the cap is lost, and once the cap is lost, XRN1 will find it an attractive substrate to cause the degradation. RPS28 protein binds to its own mRNA and recruits EDC3 to facilitate decapping. This is a kind of autoregulation. We should understand a given cell, say for example, you think of a situation, RPS28 protein is present plenty, more than, actually say if only 100 molecule are required, this cell has got 10,000. Then what will happen? Every RNA will not be able to function because every RNA has a proper poly A tail, proper cap. But what will happen? RPS28 will go and bind down to their 3' UTR, attract decapping enzyme and degrade it. So it is almost like you are drawing a line on a board. You are moving from left to right using your right hand, and your left hand with the duster you are erasing it. It will look so counterintuitive and basically a useless job. So you don't want that to happen. So who will control it? This has to be controlled by RPS28B itself. So RPS28B, if protein is there plenty, what it will do? It will go and bind onto its own RNA UTR, and then it will invite EDC3 and cause its own degradation. So once RPS28 protein is causing, or RPS28 protein is causing the degradation of its own RNA, then the protein level will decline, and protein level will decline to some extent, then other RNAs will get a chance to survive and function. And once the RNA is no more, the RPS28 level is declining to a low level, then what will happen? It won't be able to degrade its own RNA, but RNA is being constantly produced. Then the RPS28 protein will start increasing its level, because say any given cell is producing 100 RPS28 RNA, let us assume, then each RNA can produce multiple copies of this protein. Then if that much number is there, that will cause the degradation of lot of RNA. Then this 100 RPS28 RNA itself is superfluous, it is much more. Then it will cause, this protein will cause its own degradation. So the 100 number can come down to 3 or 2. You have cell has only 2 RPS28 RNA. So the protein will automatically come down. And once it comes down, then other RNA will be able to perform. But the other RNA, when it is not able to, or it is not requiring to do its job, or its purpose is done, you need RPS28. Then what will happen? The RPS28 RNA is constantly being produced. Then the degradation is now prevented. Why? RPS28 level is very low, because it already degraded its own RNA population. But now, since the production of RPS28 is going on constantly, it will build up eventually. So you can think of like somewhat like an oscillation, like a wave. It goes up, then down, again up, just like something like you can see sometimes if you watch a share market fluctuation, you can say it goes up and then comes down, again go up. It is not exactly like a wave, but trend somewhat. If market is going up now, it can come down. If it comes down, it will go up, something similar. So this oscillation has to happen in RPS28 level to provide ample opportunity for other RNA to function. Now let us see endoribonuclease mediated mRNA decay. Word itself says enough, because endoribonuclease do not bother 5' end or 3' end, whether it is protected or not protected. It can simply act right in the middle, create new 2 ends, which is 5' and 3' ends, and exosome, and the XRN1 can happily chew them up. So that is exactly what happens. Cleavage by an endoribonuclease produces 2 mRNA fragments that are susceptible to decay by exonuclease. That this 5' end is protected, this 3' end is protected. But when you cut it, you are creating a new 3' end, and you are creating a new 5' end. So the 3' end will be chopped up by exosome, the 5' end will be chopped up by XRN1. Simple. So what are the enzymes doing this? IRE1, PMR1 and RNase MRP, these enzymes can come and act right in the middle of some RNA. And remember, RNA always fight against it by forming polyribosome complex. Like every RNA is jam-packed with multiple ribosomes, then the ribonuclease are not welcome there, because there is no gap, no place. Some of them will form specific bodies. They will form like some round shaped structure and protect themselves from the degradation. But once this endonuclease have acted upon them, the fate is sealed. So MRP is name of a ribosome biogenesis that is important in the nucleus. It's contributing because processing of mitochondrial RNA also is controlled via these endonucleases. And Endoribonuclease are highly specific and are highly regulated, because we will see them. These degrading enzymes have to be controlled very effectively and very specifically. Let us see one real-time picture of mammalian P bodies. What is this mammalian P bodies? They are structures where the RNA degradation can happen effectively, and they are induced when stress is there. Like stress can come to anyone. Like if you are stressed, you are having tension, you don't sleep effectively, your body is suffering actually. So this, you don't eat food, all are stress. What happens whenever there is a stress? You can see, you can prove it. So in this study, this is culture, cell culture. This blue color is the DAPI staining of the nucleus, and the green color one is the cytoplasm. Green and red dots you can see. What is green? Green one is LSM1. We have seen it. LSM1 binds on to the 3' UTR after the decapping, and XRN1 is an exonuclease. So in a normal cell, you can see LSM1 green, which is separate, and XRN1 is also separate. Means they are not bound, or they are not in the close vicinity. That means they are there ready for action but not acting right now. But when there is a stress, you see lot of yellow parts are there, yellow dots are there. So green and red combined together, when they come so close vicinity, it will look like yellow in color. So after stress, you can see stressed condition, there is plenty of yellow bodies. That means LSM1 now attracted XRN molecule, XRN1 exonuclease, and caused the decapping. Of course, DCP1 and 2 complex would have come, and it caused the degradation. We will see more details about this RNA decay pathway in the next class. Thank you.